Good day, everyone. So we're reviewing Wetzel's Limnology textbooks. This is edition number 4, and we are on chapter 14, the nitrogen cycle, and more specifically, we are focusing on ammonium right now, NH4+. And let's get started with where we left it at yesterday. When appreciable amounts of organic matter sink into the apolymion of stratified lakes, ammonium can accumulate due to microbial activity. The accumulation of ammonium greatly accelerates as the apolymion becomes anoxic. Under anaerobic conditions, bacterial nitrification of ammonium to nitrite and nitrate ceases as the redox potential is reduced to below about plus 0.4 volts. In the sediments, a large percentage of the ammonium is adsorbed on sediment particles. With the loss of the oxidized microzone at the sediment-water interface under anoxic apolimnetic conditions, the adsorptive capacity of the sediment is greatly reduced. A marked release of ammonium from the sediments then occurs. Diffusion transport into the overlying water approximately equals production and release into the interstitial waters of the sediments. These diffusion rates of ammonium into the overlying water can be increased several-fold by the activities of benthic invertebrates such as chironomid larvae, tubiphysid worms, and bivalve mollusks. The ammonium enhancement apparently results more from water movements associated with respiratory behavior rather than from direct excretion by fauna. Excretion rates of ammonium of 4 species of chironomid and tubiphysids range from 0.33 to 2.87 micrograms of nitrogen per milligram of dry weight per day at 15 degrees centigrade. If light reaches the sediments in amounts sufficient to support benthic algae, these organisms can assimilate ammonium from the interstitial water and prevents the flux of ammonium from the sediments to the water. Because the uptake rates are in part dependent upon photosynthesis, a marked diurnal variation in release rates can occur, with reduced uptake during periods of darkness. These sources of interstitial nitrogen often serve as a dominant inorganic nitrogen source or supplement nitrogen fixation, specifically for the benthic algae and microbes attached at the sediment-water interface, particularly in nitrogen-deficient lakes and streams. These attached microbial communities can release appreciable amounts of dissolved organic nitrogen into the water above the sediments. If aquatic macrophytes, for example freshwater plants, are rooted in the sediment, these plants are capable of absorbing large quantities of nitrogen from the sediments. Much of the nitrogen can be immobilized in the below-ground rooting tissues and in the above-ground foliage tissues. Much of the nitrogen is assimilated as ammonium and can remain for long periods in living tissue and particulate detritus after senescence. Thus, in littoral and wetland areas of freshwater ecosystems, nitrogen storage in living and senescent macrophytes can be an important component. However, the nutrients stored in macrophytes can be released back to the water column again when environmental factors change, such as when plants die back at low temperatures during winter, when there is low light availability due to eutrophication, and when experiencing hydrological stress. For example, both decayed macrophytes and sediments have been shown to release a substantial amount of nutrients after dying, followed by rewetting, which might be further exacerbated by climate change. B. Nitrate and nitrification. Nitrification may be broadly defined as the biological conversion of organic and inorganic nitrogenous compounds from a reduced state to a more oxidized state. Of the numerous oxidation and reduction stages outlined in Figure 14-1, initial nitrification by bacteria, fungi, and autotrophic organisms involves ammonium plus 1/2 of oxygen is equal to 2 proton plus nitrite plus water, with a Gibbs energy of minus 276.1 kilojoule per mole. Gibbs energy of formation, kilojoules per mole, which is the thermodynamic potential that is minimized when a system reaches chemical equilibrium at constant pressure and temperature. This proceeds by a series of oxidation stages through hydroxylamine and pyruvic oxime to nitrous oxide. So we have ammonium to hydroxylamine to pyruvic oxime, or is also hyponitrous acid, to nitrous acid, HNO2. The intermediate products are labeled to physical, and heterotrophic oxidation are only found rarely in significant quantities relative to other forms of combined nitrogen. Much of the energy, total exothermic energy of minus 351.5 kilojoule per mole, released by the oxidation series is used to reduce carbon dioxide in the formation of organic matter. Detailed reactions are discussed by these authors. The nitrifying bacteria capable of oxidation of ammonium to nitrite are largely confined to ammonium oxidizing archaea and ammonium oxidizing bacteria. The intermediate products are labeled to physical and heterotrophic oxidation and are only found rarely in significant quantities relative to other forms of combined nitrogen. Much of the energy, total exothermic energy of minus 351.5 kilojoule per mole, released by the oxidation series is used to reduce carbon dioxide, CO2, in the formation of organic matter. Detailed reactions are discussed by these authors. The nitrifying bacteria capable of oxidation of ammonium to nitrite are largely confined to ammonium oxidizing archaea and ammonium oxidizing bacteria. These bacteria are mesophilic. For example, they grow best at moderate temperatures, but with a wide range temperature tolerance ranging between 1 and 37 degrees C, and grow optimally at a pH near neutrality. Oxidation of nitrite proceeds further to nitrate by nitrite plus half of an oxygen is equal to nitrate, with a Gibbs energy of minus 75.3 kilojoule per mole. Nitrate oxidizing bacteria are involved in this oxidation. Nitrobacter is the primary bacterial genus. Nitrobacter is somewhat less tolerant of low temperatures and high pH conditions that can lead to some accumulation of nitrite. The release of energy for the synthesis of organic matter by the oxidation of nitrite is much lower at minus 75.3 kilojoule per mole than that of ammonium to nitrite. The overall nitrification reaction is therefore ammonium plus 2 oxygen results in nitrate and plus water plus 2 proton. This reaction requires 2 moles of oxygen for the oxidation of each mole of ammonium. Nitrification is a key component of the nitrogen cycle in lakes. However, the recent discovery of complete ammonium oxidizers, COMAMOX, has shown that the oxidation of ammonium to nitrate can be achieved by a single microorganism. The genome of this microbe contains genes encoding ammonium monooxygenase, hydroxylamine dehydrogenase, and nitrite oxidoreductase, indicating that they have the potential for full-scale ammonium oxidation. Although conditions must be aerobic for nitrification to occur, these processes will continue until concentration of dissolved oxygen decline to about 0.3 milligrams per liter. Below this concentration, diffusion rates of oxygen to the bacteria becomes critical. Nitrification is greatly reduced in undisturbed sediment because oxygen is very low or absent. Nitrite may diffuse to the water following nitrification in the well-oxygenated superficial sediments of the littoral zone or during periods of circulation of the water and disturbance of sediments. Nitrification is severely inhibited by certain dissolved organic compounds, especially by tannins and their decompositional derivatives. Therefore, it is likely that rates of nitrification are lower in neutral or alkaline waters containing high concentration of dissolved humic organic matter. Moreover, nitrification proceeds more slowly in acidic waters such as in acid bogs and acidic bog lakes, where the pH is 5 or less. Nitrate produced in such lake is probably utilized as rapidly as it is formed, so most of the time only very low or undetectable quantities are found. Dissolved and particulate organic nitrogen. Concentration of dissolved organic nitrogen, DON, are frequently higher than dissolved inorganic nitrogen. DIN is equal to ammonium plus nitrate, with the dissolved organic nitrogen fraction often exceeding 50% of the total dissolved nitrogen pool in fresh waters, especially in highly developed catchments. In Lake Taiyu, China, average dissolved organic nitrogen concentration accounted for up to 50% of the total dissolved nitrogen. In a study of lakes and streams in Wales, dissolved organic nitrogen export from catchments was related to soil types and vegetation cover, with more vegetation conifers leading to less dissolved organic nitrogen loss. There was no seasonality in dissolved organic nitrogen concentration, reflecting the refractory nature, difficult to degrade, of many of these compounds. Many of the dissolved organic nitrogen compounds are relatively refractory. Less than 1/3 occurs as free amino nitrogen. The simple amino acids are substrates that are readily utilized by microbes. Rates of decomposition are high and results in low instantaneous concentration of free amino acids in fresh waters. As with organic carbon, the dissolved organic nitrogen of lakes and streams can be from 5 to 10 times greater than particulate organic nitrogen, or PON, contained in the plankton and ceston. The ratio of dissolved organic nitrogen to particulate organic nitrogen decreases as the lakes becomes more eutrophic, are closer to 1:1 in the trophogenic photic zone, and increase in the tropholytic aphotic zone. More organic nitrogen is typically synthesized by small phytoplanktonic algae, less than 10 micrometers per unit cell volume, than by larger forms. A significant portion of algal intracellular nitrogen, 10 to 20% in the Cyanobacterium oscillatoria, is released extracellularly, mainly as protein and ammonium nitrogen, with smaller amount of nitrite and amino acid nitrogen. Bacterial utilization of these organic compounds, particularly the amino acid, is extremely rapid and linked with organic carbon availability. Different types of bacteria have been shown to have preferences for different dissolved organic nitrogen forms. For example, one study found bacterioplankton groups had distinct preferences for a range of low molecular weight organic compounds. Algae, and especially cyanobacteria, also excrete polypeptides and other organic compounds that are capable of forming complexes with metals such as iron and copper, as well as phosphates. These complex organic compounds differ in their solubility and physiological availability. Similar nitrogenous compounds have been found to be secreted by larger aquatic plants. In some situations where the littoral zone is extensively vegetated, the release of dissolved organic nitrogen by microphytes can form a major source of organic nitrogen for the lake. Furthermore, as aquatic vascular vegetation decomposes, large quantities of organic nitrogen are released. Much of this organic nitrogen is absorbed by the sediment and is utilized by attached macrobiota, where decomposition can rapidly become limited by inorganic nitrogen, especially under anaerobic conditions. There is limited information on which forms of dissolved organic nitrogen are available to phytoplankton or bacteria. However, it has been established that many species of algae from diverse groups are able to utilize urea, simple amino acids, and a range of other simple nitrogen compounds such as nitrogen source. Among the algae of Lake Biwa in Japan, for example, relative assimilation rates were highest for ammonium, average of 74% of total, followed by urea, an average of 20%, and then nitrate, average of 6% of total. A study of the green algae Rhaphidocelis subcapitata found that a wide range of bioavailable compounds such as amino acids, urea, DNA, and RNA, as well as some humic acids, could be used for growth. The filamentous cyanobacteria, for example, Planktothrix rubescens, the Macrocystis complex, and Spirulina platensis are known to assimilate several low molecular weight organic compounds. Conversely, at higher concentration, the amino acid proline and arginine have been found to inhibit cyanobacterial growth. Catchment scale nitrogen budgets. One way to quantify inputs, outputs, and retention of nitrogen by freshwater system is to use nitrogen budgets where all sources are accounted for. The quantification of nitrogen inputs and outputs for rivers and streams is challenging at the watershed scale, so it is normally estimated using hydrological and water quality models, including simple export coefficient models, statistical models, mechanistic models such as SWAT, and hybrid mechanistic statistical models such as Sparrow. Terrestrial sources are the main nitrogen sources of rivers and streams during storm events. Groundwater nitrogen deliveries can be significant, especially when groundwater is enriched with nitrogen via leaching and infiltration from surface soils, especially when fertilizers are applied to soils. Studies also showed that atmospheric deposition can be a significant nitrogen source in some rivers, with nitrogen fixation, use of nitrogen gas by organisms for growth, section 3b, being important in some lentic systems. The output of nitrogen is mainly through denitrification, anamox, that is anaerobic ammonium oxidation, and export to oceans. According to a review of nitrogen removal in streams of agricultural catchments, in stream processing can remove 10 to 70% of the total nitrogen load to drainage networks at the watershed scale on an annual basis. Maringer and al. Made global estimates of nitrogen inputs and outputs to and from freshwaters and determined that the major nitrogen input to freshwater was from catchments inputs, with atmospheric inputs being approximately 10% of this. Approximately 45% of inputs from runoff and the atmosphere were lost to the atmosphere, with only 15% buried and the remainder exchanged at the mouth. Residence time was shown to play a critical role in the processing of nitrogen, phosphorus, and carbon through the riverine pipe, where longer water residence times from streams to lakes result in substantial increases in the carbon-nitrogen, carbon-phosphorus, and nitrogen-phosphorus ratios. Nitrogen removal through anamox, to be discussed in this chapter, has also been shown to be important in turbid river systems. Nitrogen budgets at a global scale can also be used to highlight the impact of anthropogenic activities. A comparison of pre-industrial nitrogen inputs and outputs from catchment showed that annual fluxes have increased substantially. So let's look at this table and this figure here. So first the table. So table 14-4, estimated of carbon, nitrogen, and phosphorus terrestrial inputs, atmospheric inputs, and losses, reservoir processing and losses to burial through the freshwater pipe, as well as export to the coastal ocean. So it's in teragram per year. So we have catchment inputs, we have dissolved organic carbon, particulate organic carbon, dissolved inorganic carbon, nitrogen, phosphorus, the carbon to nitrogen ratio, the carbon to phosphorus ratio, the nitrogen to phosphorus ratio, the carbon, nitrogen, and phosphorus ratio. We have catchment inputs, atmospheric flux, total burial, coastal exports, and reservoir processing. So what jumps at us here? let's look at the high and the low numbers. So catchment inputs, what dominates is the dissolved inorganic carbon. And we can see that definitely like carbon dominates, 375 to nitrogen to phosphorus. So phosphorus is probably the laggard in this whole budget between those three elements. Atmospheric input, so we have input, output, and net. Inputs, we have particulate organic carbon, we have nitrogen, and then phosphorus. Output, we have dissolved organic carbon, particulate organic carbon, and then some contribution from nitrogen. And so once again, carbon dominates. Total burial, so we have particulate organic matter that dominates, some amount of nitrogen burial as well. And definitely carbon dominates in terms of burial. Coastal exports. Coastal export mostly is dominated by dissolved inorganic carbon, some particulate organic carbon, as well as dissolved organic carbon. And let's see, we have carbon to phosphorus once again, very strong discrepancy between the two. And reservoir processing, and that is dominated by particulate organic carbon, and in second hand by dissolved inorganic carbon. So there's something opposite between coastal export and reservoir processing here. So where we have coastal export dominated by dissolved inorganic carbon, and then we have reservoir processing dominated by particulate organic carbon. And let's look then at figure 14-3, changes in the global nitrogen budget from contemporary to pre-industrial periods. Numbers represent global land nitrogen storage in teragram of nitrogen, or annual exchange fluxes in teragram of nitrogen per year for contemporary 1991 through 2005 and pre-industrial between 1831 and 1860. Average in temperature, in parentheses, times. Okay. So we have, let's see here. So we have the values, the pre-industrial values in parentheses, and then we have the values between 1991 and 2005 in without the parentheses. So freshwater emission, we have about twice post-industrial compared to pre-industrial. Atmospheric depositions, it's about a little less than 3 times. Fertilizer application, we have actually very high. Like pre-industrial, we didn't have any, and then post-industrial, we have very high. So this is where the industry helped us create these fertilizers. Harvest emissions, so we have about 3 times more in post-industrial times. Soil emission, that's really like there's a big difference. This is like about 6 times more in post-industrial times. Fire emissions, it's about the same, a little less in pre-industrial time. And biological nitrogen fixation, somehow humans have been able to influence that. So we have about 1.5 times. Vegetation, so vegetation is about the same. Rivers and lakes, we have twice more post-industrial compared to pre-industrial in terms of nitrogen being present in that pool. And then rivers, we have about 1.5 times. Unit in teragram per year, and unit in storage in teragram of nitrogen. And that's going to be about it for today, and I'll see you tomorrow, and tomorrow we'll pick it up with nitrogen sources, transformations, and fate in lakes and reservoirs. That will be it for today, and tomorrow we'll pick it up with sub-chapter 2, nitrogen. That will be it for today, and tomorrow we'll pick it up with nitrogen sources, transformations and fate in lakes and reservoirs. Thank you and see you tomorrow. Bye.